Welcome back to Abnormal Psych. Today we'll talk about multidimensional approaches, some very basic groundwork for a biological approach, and apply this in a few cases, including the case of what it means to say something is caused by genes. So let's make like an amateur TED Talk and start with a straw man, a one-dimensional model. Imagine someone who thinks that what they focus on, what they understand, what their theory is, the singular dimension about which they know, explains abnormality, mental illness. In other words, all they need is that one perspective, one dimension. Learning, mental illness, behavior change, it's all conditioning. It all depends on what you've been rewarded for and punished for. I like to personify models and theories as little hedgehogs. Most of you are probably familiar with the fox versus hedgehog distinction. Hedgehogs are experts who are really good in one model, one dimension. They know a whole lot about their field. They're dug very deeply into it. And foxes are people whose knowledge spans a whole bunch of different areas. So they're quite clever, but they don't have the depth of a hedgehog in any one of these areas. We need both types of thinkers, but in general it's better to be a fox. So we can contrast one-dimensional models or hedgehogs who adhere to them with a multidimensional model approach, or the foxes who cultivate them. So the fox with her multidimensional approach might walk up to a problem and say, well, this person clearly has a unique biology that has been shaped in part by the learning that his biological system has done, some of which was emotional learning, and this affects his thoughts when he goes to make decisions, especially decisions that might be analogous to or related to the social context in which he did his pathological learning, and moreover, he probably only acts his pattern out because there's a cultural expectation for him to do X or to not do Y. In other words, there are tons of relevant perspectives or levels of analysis here, and it's not just that they all contribute useful information, it's that the levels or dimensions being referred to interact with each other. This makes a multidimensional approach complex because each dimension or theoretical perspective you add creates numerous interactions between that dimension and the other dimensions you had previously. In other words, multidimensional thinking or theorizing is exhausting. But that makes it analogous to reality, doesn't it? We're not just cells in a petri dish. We're not just social performances. We're not just a list of a few emotions, or a few cognitive schemas, or a series of pre-wired conditioned responses. But we are all of those things bundled into one. Difficult but not impossible to predict mess. So multidimensional models, by virtue of admitting, or at least trying to capture, all of these sources of influence on the individual, are more representative of reality. Doesn't make them more true by definition. A one-dimensional model could be more strictly true than a multidimensional model. Can you engage in a behavior that has not been learned? Well, no, not really. Can you have a thought that does not rely on biology? No, you can't. So one-dimensional models can often seem like they have everything covered. But hopefully you'll come to agree, as we progress through the course, that you just haven't done your due diligence as an intelligent person if you've only analyzed a problem through a single lens or along one dimension. To identify the causes of various psychological disorders, we must consider the interaction of all relevant dimensions: genetic contributions, the role of the nervous system, behavioral and cognitive processes, emotional influences, social and interpersonal influences, and developmental factors. Is that an exhaustive list? I'm not sure what could be, but that's a good first draft. For a multidimensional integrative approach, you need at least 3 dimensions to cover your bases. You need the biological, the psychological, and the social level of analysis. This diagram splits the psychological level into blue and orange here, emotional and cognitive influences and behavioral influences. But of course you could easily split orange up into classically conditioned versus operant behaviors. And you could easily split blue, emotional and cognitive influences, into hot versus cold cognitions, i.e. you could put emotions in one bin and cognitive in the other. So we're splitting, right? If you're a lumper, then you'd prefer biopsychosocial. 3. You've lumped them into as few lumps as possible with 3. This image gives us 4. You could make a good case for 6, but this is starting to tax my working memory. I kind of want to lump them back again into biopsychosocial. So let's consider a case so that we can see how we would use this multidimensional approach. Let's consider anorexia nervosa. First off, if you open up the DSM 5th edition, you will see this as requirement for diagnosis of anorexia nervosa: restriction of energy intake relative to requirements leading to a significantly low body weight in the context of age, sex, developmental trajectory, and physical health. Significantly low weight is defined as a weight that is less than minimally normal, or for children and adolescents, less than that minimally expected. You can't get the diagnosis without the low body weight. Criterion B, you need all of the letter criteria: intense fear of gaining weight or of becoming fat, or persistent behavior that interferes with weight gain, even though at significantly low weight, such as going out and running 10 kilometers despite the fact that you are malnourished and your body probably cannot spare the calories required to run that 10K. Finally, self-perception issues: disturbance in the way in which one's body weight or shape is experienced, undue influence of body weight or shape on self-evaluation, or persistent lack of recognition of the seriousness of the current low body weight. That's what it takes to get a diagnosis of anorexia nervosa. Now let's consider a case so that we can consult our model or demonstrate how our multidimensional approach would work. Meet Sarah. Sarah is an adult female with a body mass index of 15, mild anemia, and low bone mineral density. So she's not keeping up with what her body needs. Despite her weight, she diets out of fear of becoming unattractive. So we have 2 of the 3 criteria covered. Finally, she recognizes her health problems, but insists that keeping her figure should be the top priority of therapy. That's going to be difficult considering how distorted her self-perception is. So let's make like a fox, and let's go to a bunch of hedgehog holes, and we'll talk to a bunch of different hedgehogs. Our first hedgehog says, "Listen, I'm the expert in genes, in the innateness of behavior and propensities, and let me tell you, anorexia nervosa, like everything else, is all about genes. And let me tell you why. First off, you've got what we call family resemblance. If you have a sibling or a parent with anorexia nervosa, you're more likely to get it yourself. That points to genes. And that can't just be explained away by the fact that first-order relatives tend to have similar values and contexts, because if you have an identical twin with anorexia nervosa, you're more likely to have it yourself than if you had a fraternal twin with anorexia nervosa. So these 2 types of twins share just as much context. What the identical twins have more of is genes in common. So you see, take it from me, the bio hedgehog, anorexia nervosa is all about genes. There's also studies from the US that show individuals from different ancestral groups have different rates than others. Presumably it's the difference in genes between the groups that explains this. Moreover, it might have something to do with hormones or sex chromosomes, because females are 10 times more likely to have anorexia nervosa than males. Okay, very good. Thank you for your presentation. Bio hedgehog. It was very good, but we are a fox. We're not easily convinced by things. We're not a donkey that gets led around by any hedgehog that can put together a PowerPoint presentation. Let's consult a couple more hedgehogs. The psycho hedgehog will tell you that anything, including anorexia nervosa, is all about psychology or personality, and the psycho hedgehog will have plenty of evidence. First off, says psycho hedgehog, personality traits predict, and in modeling, contribute to anorexia nervosa. Not only this, but the decisions that your personality or particular psychology leads you to make work to amplify those traits by getting you to choose situations that feed into and amplify those traits. So if you are focused on your body image and your weight, then you're going to get along better with friends who are also weight focused. And this might even become, by virtue of the choices you made, which were driven or motivated by your personality, or your traits or your personal preferences. These could even determine your choice of profession: athlete, model, influencer. So you see, says the psycho hedgehog, you start off with a preference or a propensity, a rather minor trait, but the choices that this trait leads you to amplify that preference, leading in some cases to the extreme of anorexia nervosa. Okay, very good, we say, but we are a fox, and we demand at least one more hedgehog to spend her valuable time explaining things to us. Let's look at the social hedgehog. It's all culture and society, she says. First off, we don't really see anorexia nervosa as a problem in places that are not what we call post-industrialized as societies. Sure, we see it in Canada, Japan, the United States, Australia, but how many people are talking about anorexia nervosa in less quote-unquote developed places? In other words, says our social hedgehog, anorexia nervosa is a symptom of post-industrial angst or affluence or anxiety or capitalism or consumerism, definitely some isms. And the social hedgehog would also have a different take on a fact that we saw utilized by the psycho hedgehog. Namely, anorexia nervosa is more common in occupations with a physical focus. So the psycho-level hedgehog said, you have people with certain traits and preferences going into these occupations. But our social level hedgehog is free to posit that maybe those occupations cause people to have anorexia nervosa. So now we've consulted 3 hedgehogs, and we, being a fox, get to put them together. Now, we, being a fox, know that the foundational premise of each of the 3 hedgehogs, one dimension can explain everything, is false, is inadequate or incorrect. So the bio-hedgehog says it's all genes and predispositions. The psycho-hedgehog says it's all psychology or personality. And the social hedgehog says, well, it's all culture and society. What makes them wrong? Just the word all. We, as a fox, would conclude that because each hedgehog ignores the other, they are, at least in that limited respect, wrong. Here are our bio, psycho, and social facts. Note that you could draw lots of arrows between the different facts. So these 2 are probably related. These 2 are pretty much the same fact. And there could be some cultural values explaining the differences by ancestry. So what the fox would view as correct then is the statement it's genes, psychology, and culture/society together, but the fox would add it is these things together interacting, influencing each other. So taking the fox multidimensional perspective involves more than just removing the word all from the strawman one-dimensional hedgehog examples. It also involves adding in admitted complexity in the form of all of these different explanations, make reference to things that interact with each other. In other words, all of these different causes of anorexia nervosa need each other in order to get anorexia nervosa from them. Where do your personality preferences come from? Genes. Who is selecting their way into different occupations? The people making decisions to join them. And maybe social level identification with a group leads you to conform to different expectations, or Latina female, etc. Let's strip this down. Why did the chicken cross the road? Bio Hedgehog knows the answer. It's because the chicken's brain is wired for exploration. If it didn't cross the road, its ancestors wouldn't have survived. So it crosses the road. Psycho Hedgehog knows the real answer. It's because that particular chicken has an adventurousness trait. But don't discount the social level hedgehog. She says that something about the other side of the road is better for that particular chicken. Next, neurons and neurotransmitters. First, a useful metaphor to get us roughly on the right page. Just like a computer does everything with ones and zeros in terms of information, so our complex brain does everything with firing or not firing nerve cells or neurons. On some level, or on one dimension, the complexity of you comes from just a whole bunch of firing versus not firing neurons, which you'll recall are just cells that communicate with each other. You have around 85 billion of them, which make roughly 160 trillion connections with each other, i.e. synapses. The cell body of the neuron is constantly making material like neurotransmitters and the protein required to make more synapses, more connections. Most of these synapses go on the dendrites, which receive most of the signals. Though there are some synapses on the axons, which generally are just responsible for carrying or propagating an electrical charge along their length until that signal, or action potential, reaches the axon terminals, at which point, mostly due to an influx of calcium into the cell, or at least calcium ions into the cell, the electrical signal turns into a chemical one, i.e. neurotransmitters are released into the synaptic cleft, which is the space where synapses work, i.e. information in the form of chemical messengers is transferred. Here are some neurotransmitters being released from their vesicles, from the axon terminals into the synaptic cleft. The neurotransmitters are released from the presynaptic neuron, and then they make contact with the postsynaptic neuron's receptors. They do not, for the most part, enter the postsynaptic cell. They simply allow for the opening of ion channels, changing the charge of the postsynaptic neuron. sometimes propagating the action potential, i.e. causing the postsynaptic cell to fire as well, but sometimes causing hyperpolarization of the postsynaptic cell, i.e. making it less likely that that postsynaptic cell is going to fire. This is what your brain cells are doing right now. In fact, they never stop. When your brain is at rest, it is still using 20% of your burned energy just to stay at the ready, i.e. to maintain negative charge in the neurons, and if you're fully relaxed, to run what's called a default mode network, the bit of you that's on when it seems like nothing else needs to be on. Your brain is an expensive piece of hardware in terms of the energy that it takes up. But of course your brain has more cells than just your neurons. The performance of the neurons and the upkeep of the neurons relies upon other cells, like those that comprise the myelin sheath, the fatty tissue in which a neuron is wrapped, along the axon to help speed propagation of electrical signals within its cell walls. In the peripheral nervous system we have Schwann cells, and when these cells aren't properly myelinating your axons, you have the demyelination disorder of Guillain-Barré syndrome. Essentially, some of the signals are a little bit off in terms of speed and coordination because of the demyelination, and so you experience pain and muscle weakness, among other symptoms. When demyelination occurs in the central nervous system, an example of this is multiple sclerosis, where you similarly experience weakness, a loss of coordination, and oftentimes a loss of sight as well. In the central nervous system, you have the glial cells, helping to form the myelin sheath, and helping neurons to wire up together and to coordinate their maintenance in terms of waste removal and access to blood oxygen. Glial cells got their names from the word glia, which means glue, because we thought the neurons were the stars of the show, and the glia just held them together. But not only do we have as many glial cells as we have neurons, it turns out that they help with organization, coordination, and maintenance of the neurons. So the two types we have here are astrocytes and oligodendrocytes. The astrocytes, named because they look like a star, can connect tons of neurons together. They can help the neurons find each other. The presence of astrocytes indicates usually an increased reliability of neuronal transmission. And the feet of the astrocytes help to line the blood vessels in the brain to comprise what's called the blood-brain barrier, to help your brain be safe from or not poisoned by large or charged molecules. So while they are helping with the blood-brain barrier, the oligodendrocytes are helping make the myelin sheath in the central nervous system, providing the neurons what they need to maintain themselves and to optimize growth and transmission. So a neuron could be firing or not firing. When a neuron is not firing, it will maintain a negative charge and be ready to fire, and this is called the resting potential. Maintaining this resting potential is why your brain takes 20% of your resting energy if you're just sitting around. So you can see in our little picture of a neuron that there are many places, many dendrites with synaptic connections, at which a given neuron could be receiving signals. Some of those signals could be encouraging firing, and some could be discouraging firing, i.e. letting negative ions into the cell. Whether all of the given input into a single cell body amounts to the cell firing or not firing is something that is determined at what is called the axon hillock, the start of the axon. If the sum total of ionic input amounts to enough positive charge, once everything meets at this axon hillock, then an action potential will be propagated down the axon. But if enough negative ions enter to counter the push for positive charge, then an action potential won't make its way from the axon hillock down the axon. The action potential is triggered by positively charged ions rushing through the axon hillock, opening sodium ion channels all the way down, until at the end, at the axon terminals, most of the positively charged ions that are entering are calcium, which the postsynaptic cell can often make good use of in terms of second messengers. This is an illustration of how the resting potential is maintained. We call it the sodium-potassium pump. How do you maintain a negative charge in a cell? You nearly constantly trade 3 positively charged ions for 2 positively charged ions. You spit out 3 sodium ions, which is one of the reasons why when you have an action potential, the positive ions that are flooding into the neuron are mostly sodium. The neurons just spit them out there, so they were available. And what gets transported in is 2 potassium ions. This maintains the negative charge, the resting potential, and we don't need to worry about having too negative of a charge because the cell would then just leak. An example of us messing with this system, let's say you're experiencing manic states and you need something to slow you down or even you out, we might prescribe lithium. The lithium ions tend not to get transported out of the cell, so the cells don't maintain the resting potential as efficiently or as over-efficiently as they previously had. This can reduce neurotransmitter effectiveness or reduce signal or action and work to stabilize mood. This is pharmacotherapy at the level of ion. So the firing of a neuron can then seem like quite a big process, but this firing and resetting occurs so quickly that a typical neuron can fire 200 times within a second. Shorter neurons can fire faster since they require less time to propagate the signal and less time to restore their resting potential. So that referred to communication inside the neurons, which is electrical, the propagation of a positive charge, but communication between neurons, i.e. across synapses, is chemical, affected by neurotransmitters or pharmacological agents that mimic them. So when someone is making reference to a quote-unquote chemical imbalance in the brain, they're talking about neurotransmitters and their action. Each neurotransmitter is limited in terms of the receptors it can activate. The analogy or metaphor here is lock and key, i.e. the chemical structure of some neurotransmitter molecules happens to work with the protein structure of some receptors to allow positive or negative ion channels to open, thus increasing or decreasing the charge of the postsynaptic cell. Now, the neurotransmitters would be fine with just hanging out in the synaptic cleft and jumping from receptor to receptor and continuing to activate them. But some of the neurotransmitters end up getting broken down, and many of them end up getting reuptaken into the presynaptic cell from whence they came, a process called reuptake. We noted that some neurotransmitters tend to excite while others tend to inhibit the postsynaptic neuron, and each neurotransmitter has a kind of profile in terms of its effect on the postsynaptic cell. So let's take a few neurotransmitters and see what roughly they do. Glutamate is in all of your cells. Every neuron has it, making it the most common neurotransmitter in the central nervous system. It, being excitatory, is complemented by GABA, which is inhibitory. In other words, glutamate released into the synaptic cleft is going to attach to postsynaptic receptors and tell them to let positively charged ions into the postsynaptic cell, working to activate or to make that postsynaptic cell fire. GABA, when released into the synaptic cleft, is going to make the postsynaptic cell receptors let negatively charged ions into the postsynaptic cell, thus inhibiting firing. Recall that whether the postsynaptic cell ends up firing is going to depend on whether the positive and negative charges from all of the different dendrites end up at the axon hillock overall positive or negative. Most anxiolytic drugs, i.e. most drugs that work to calm your mind without actually putting it to sleep, work at some point to promote activity of the GABA neurotransmitter. GABA, which is short for gamma-aminobutyric acid, calms the brain. It says, shh, it's going to be okay. Next, we have acetylcholine, which does a lot of work in the central and peripheral nervous systems. It is the endogenous neurotransmitter that nicotine is mimicking when it picks you up a bit and helps you focus. In fact, many of the receptors that stand between your neurons and your muscles, i.e. the connections that turn your thoughts into muscular movements, are realized at nicotinic receptors, at what's called the neuromuscular junction. Next, the family of monoamines, norepinephrine, dopamine, and serotonin. Norepinephrine in the blood, where it's called noradrenaline, works with the norepinephrine that's released as a transmitter in the central nervous system to mediate the physiological changes that make up the fight or flight response. So norepinephrine can elevate blood pressure and heart rate, decrease your digestive activity, dilate your blood vessels to feed your skeletal muscles, your heart, and your brain. In the central nervous system, the tasks of norepinephrine include moderating your arousal, keeping your cognition on track, and consolidating memories, especially memories of emotional experiences. A more specific example of keeping your cognition on track would be some studies done with rhesus monkeys that showed that increasing available and active norepinephrine improved their performance on a delay and distractor test. So the monkeys see something, like food, that they want hidden in a given place. But before they can go get it, there's a period of waiting. That's the delay. We may also have, in this intervening waiting time, some things that might distract the monkey from remembering where the food or treat that he wanted was hidden. More norepinephrine activity, that is, pharmacologically enhanced levels of norepinephrine, reduced and nearly eliminated the effect of the distractions. In other words, it helped to keep them on task. There are different types of receptors in the brain for norepinephrine. Under normal conditions, what you want for optimal arousal and cognition is for norepinephrine to facilitate prefrontal cortex function by activating the alpha-2 adrenoreceptors. But one thing that facilitates norepinephrine activity is stress, and if you have too much stress, you have too much norepinephrine activity, which in the brain is not just your alpha-2 adrenoreceptors being activated, but also your alpha-1 adrenoreceptors being activated as well. In normal conditions, your norepinephrine was keeping you on task and keeping you just aroused enough to do a good job, like the rhesus monkeys. But under stress, when you have too much norepinephrine activity and your alpha-1 receptors are working, the norepinephrine leads to cognitive impairment. You need some stress and some investment in order to activate the norepinephrine that's going to keep you on task, but too much is going to impair that working memory boost that just enough was giving you. In other words, cognitive functions are optimized at an intermediate level of norepinephrine-induced prefrontal cortex activation. Next, monoamine, dopamine. Dopamine handles things as simple or basic as coordinating motor movements, and as complex, profound, and cosmic as finding meaning in life. Pretty much everything that is rewarding subjectively at some point involves the dopamine circuit, including the nucleus accumbens. Funny jokes give you a shot of dopamine. Doritos, even though they don't really taste like anything, give you a shot of dopamine. Watching a 7th episode of a show on Netflix when you're supposed to be doing something else gives you a shot of dopamine. Too much dopamine can make normally insignificant things seem meaningful. There are different versions of the dopamine hypothesis in schizophrenia. We'll take a simple one here and we'll say one of the problems in schizophrenia is too much dopamine. So dopamine tells you what's important, what's interesting, by giving you a reward for looking at it. If you're getting too much dopamine, you are going to see too many things as important. In schizophrenia, one of the things you may see as important that shouldn't be important are basic internal speech fragments or partial activations of things you might have said, or commands that wouldn't normally have made it all the way into your conscious mind. A dysregulated dopamine system says, wait, what was that thought? What was that thought? What was that voice? What did I just hear? And so the person suffering from schizophrenia has the volume turned up on these unimportant thought fragments, which ends up looking like a very disorganized mind that perhaps attributes some of its thoughts to other people, spirits, voices, conspiracies. So nearly every psychopharmacological intervention, nearly every medicine that we give people for schizophrenia, works to block or reduce dopamine transmission. An issue with this is, if you prevent dopamine transmission too effectively, you end up with Parkinsonian symptoms, i.e. you lose your ability to effectively move your body. The flip side is, individuals with Parkinson's disease often benefit from pharmacological interventions that increase their dopaminergic activity. And if we give these individuals too much dopamine, they can have medication-induced symptoms of schizophrenia. A particularly strong schizophrenia medication is haloperidol, which is sometimes used for those schizophrenia patients with a history of violence because its effects are somewhat tranquilizing. It blocks dopamine receptor subtype 2, and at high doses, it can induce catalepsy, i.e. a lack of movement. At lower doses, in humans and animals, it will just suppress what we call exploratory behavior. It will make you uninterested in things so that you do not do anything. Because if dopamine is blocked, meaning and appreciation are blocked. Our third monoamine, serotonin, the rest or aggress neurotransmitter, which plays a sizable role in sleep, anxiety, and aggression. The most common behavioral effect of eliminating serotonin in the brain is an increase in aggressive behavior. The serotonin deficiency hypothesis of aggression says that low central nervous system serotonin activity makes people overly aggressive. Some personality disorders are associated with violent behaviors, and in these cases, we might consider pharmacotherapy for these personality disorders, and we call the serotonin boosters that we would give these individuals serenics, root there being serene. SSRIs would be one candidate for this type of treatment, i.e. treatment to reduce hyperaggressiveness, because SSRIs, selective serotonin reuptake inhibitors, keep serotonin in the synaptic cleft, so that the neurotransmitter molecules can continue moving from receptor to receptor on the postsynaptic cell, leaving your brain to think that it has more serotonin than it actually does, because in effect it does, helping you in this case to reduce your aggressiveness. But of course SSRIs are used for other things too, like improving sleep, and over longer terms, think weeks or months, SSRIs can lead to neurochemical changes that reduce anxiety and depression as well. But serotonin does far more than this. In fact, most of the serotonin in your body is located in your gut. Serotonin is what, when you ingest food, tells your intestines to engage in peristalsis, moving the food in your gut around, and to engage in secretion. So if you're having some psychiatric symptoms, and maybe we've tried more targeted approaches, like a dopamine blocker or an SSRI or an SNRI, selective norepinephrine and serotonin reuptake inhibitor, but those more targeted approaches didn't work, we might take a more shotgun approach and just try to give you more of all of the monoamines by preventing something called monoamine oxidase from doing its job. Monoamine oxidase works to break down monoamine, so this regulates how much you're going to have in your system, or specifically in each cell. If I give you a monoamine oxidase inhibitor, then you will be breaking down norepinephrine, dopamine, and serotonin less, so you'll have effectively more of it in your system, improving your arousal, reward, and rest, digest, and egress activity. And that all seems great, until you realize you don't necessarily want more of these things, because you don't want cognitive impairment or schizophrenic symptoms, so psychopharmacotherapy is a tricky venture. If you've ever been put on meds for something before, you've probably gone through a trial and error stage in terms of dosage, medication type, until you finally landed on what helps. Next, neuropeptides, specifically endorphins, which is short for endogenous morphine, which we're pretty sure exists in order to help you manage your pain. Now, it's unlikely that you have to run away from predators today, or that you will have to run after prey as a predator today. But back in the day, having endogenous morphine that became active when you engaged in strenuous tasks that cause you pain, like running long distances, helped you accomplish those tasks and survive and thrive. If you've ever experienced a runner's high, the pleasure that comes from physically pushing yourself, a kind of wave of intense satisfaction, then you've gotten high on your own supply of endogenous morphine. The receptors for your endorphins are what exogenous opioids, like morphine and fentanyl, activate in order to give you a high, or what something like OxyContin is designed to do, which is to prevent pain. Anandamides have an entirely different function. An example is your endogenous cannabinoids, which, if you've ever experienced a cannabis high, you have a pretty good idea of what your endogenous cannabinoids are doing. These get you to eat, these make you motivated, they help with memory, and they help with sleep. These are the systems that get dysregulated when you use marijuana regularly, hence the stereotype of the demotivated stoner. One thing that can happen if you are low in endogenous cannabinoids is you can be more sensitive to stress-induced anxiety. The cannabis withdrawal symptoms should provide ample examples of what endogenous cannabinoids do, because the cannabis withdrawal symptoms occur largely due to your use of cannabis having dysregulated your endogenous cannabinoids. In other words, your body got used to exogenous cannabinoids being introduced and stopped creating its own. So now that you stopped bringing in exogenous cannabinoids, you are deficient in cannabinoids. So the withdrawal symptoms show you what your normal, self-created endogenous cannabinoids normally handle. What are the typical withdrawal symptoms for cannabis? According to addictions research, irritability, anger or aggression, nervousness or anxiety, especially stress-induced anxiety, difficulty sleeping, decreased appetite, restlessness, depressed mood, and finally a catch-all for physical symptoms, pain, tremors, sweating, headache, chills, and fever. Up with sleep difficulty should be weird dreams. My colleagues in addictions say that the two typical cannabis withdrawal symptoms that they tend to see in their clients that distinguish themselves as cannabis withdrawal symptoms are bizarre dreams and abdominal pain. Anxiety and irritability are more common, they're just less distinguishing because so many other things can cause irritability and anxiety. So those are a few types of neurotransmitters. When you have psychoactive drugs, you have things that act like these neurotransmitters. Exogenous things from outside the body that once you put them in your body, fool your body into thinking it has some of these types of neurotransmitters. If a psychoactive drug tricks your body into thinking you have more of an endogenous substance, like serotonin, then it is an agonist. So a selective serotonin reuptake inhibitor is a serotonin agonist. It increases serotonergic activity. An antagonist would be something that decreases the activity associated with a given neurotransmitter. We mentioned dopamine blockers for schizophrenia, like haloperidol or chlorinated promazine. Now, I mentioned that if we want to affect a change in depression or anxiety, selective serotonin reuptake inhibitors take weeks or even months to have any major effect when they work. This is because it's not just availability of serotonin, but larger neural changes that occur over time as a result of the increased availability of serotonin that help with these symptoms. This is one example of neuroplasticity, or changes in what your neurons are doing, or the very structure that you could say a group of neurons make up. Your brain has amazing power to change. Simple learning is neuroplasticity. If you can remember something that happened to you yesterday, that is a change in your brain. You didn't have that memory the day before yesterday. Your brain changed to have it so that you have that memory now. It might not have it forever, but that's because the brain changes. So learning, like the type of learning you do in a course, is in part the creation of new synaptic connections. You have to build on what's there, but hopefully you can connect things up, make new effective firing patterns that constitute new learning, new associations. Now this is a skill or physiological effect that decreases a bit across your adulthood, like so many other things. For example, the time it takes you to heal cuts. But a brain is plastic as long as it's alive. You're always learning. Pop quiz. Which of these types of psychoactive drugs promotes neurotransmitter activity? It's agonist. Next, your nervous systems, because these provide so many of our metaphors for understanding things, especially anxiety. Your central nervous system is your brain and spinal cord. It takes in sensory information and produces things like action and decisions, while your peripheral nervous system handles things like turning those decisions into movement and performing all of the tasks that keep you alive that you don't have to think about, like beating your heart. Imagine if you had to consciously make your heart beat, like you consciously open and close your hand. Now imagine that you could will your heart to stop beating. You can't, but dolphins can. Go ahead and Google it. There's been plenty of cases of dolphins that have just decided to stop living by shutting off their hearts. That's not an option for Homo sapiens sapiens. The beating of our heart is controlled by our autonomic system, part of the peripheral nervous system. Sympathetic activity can increase the heart rate until the parasympathetic system comes in to calm it down and do some basic maintenance. So here are the nerves that are outside of your brain and spinal cord. The somatic ones handle willed actions and reflexes, and the autonomic nerves dilate pupils, control glands, and they handle automatic responses to the environment, like the fight or flight response in the sympathetic branch, or the rest and digest action of the parasympathetic system, which says it's time to rebuild parts of the body, handle the basic processes you need to survive. Importantly, when your emergency system, the sympathetic system, is active, your parasympathetic system is suppressed. So the more you are in emergency mode, the less you are handling the basic maintenance of your body, like creating more cells to fight infection, healing from wounds, digesting your food efficiently, which is one of the reasons why you're more likely to both feel and get sick when you're under more stress, or when you're engaged in prolonged worry or anxiety. Your endocrine system handles things like the secretion of hormones, chemical messengers, and other things that help to regulate or dysregulate your emotions. The pituitary gland plays an important role in signaling hormone-releasing glands. It can be viewed as the connecting bit between the mind/brain and the body, because it controls glands like your adrenal glands, but it's controlled by the hypothalamus. Giving you an axis, you have the HPA axis, hypothalamus, pituitary, and adrenal axis. Signals from the pituitary are responsible for the release of hormones. One of the most important effects of these hormones would be the regulation of your blood pressure and the release of hormones and neurotransmitters like oxytocin, the presence of which is associated with bonding and love, especially in females, and especially when those females are breastfeeding. Moving down the HPA axis to the adrenal glands, which are sat atop of the kidneys. Here, sympathetic activity can release adrenaline and the stress hormone cortisol. The effects of the adrenaline will be that you have increased energy production in your muscle cells, so you can do things like fight or run, which of course takes energy from other processes, like digestive processes. And it's cortisol that's going to have the blood pressure effects. And like a good evolutionary survival mechanism, cortisol says if you are under stress, then you are probably being attacked or starving, or something else life-threatening is probably happening to you. So I'm going to increase your blood pressure so you can do things like run and fight, and I'm going to make sure you store all the available food that you intake, because if the emergency that your body is apparently experiencing happens to be starvation, we wouldn't want to excrete anything that your body could possibly use, so I'll store any carbohydrates that you don't need, any fats that you don't need immediately, on your midsection, which is one of the reasons why stress leads to adipose tissue around your midsection, because cortisol thinks that anything that's stressing you out deserves the same response as potential starvation. Evolutionarily adaptive, not so much necessary today here in Canada. How deleterious or how damaging is sustained cortisol release or constant sympathetic activation, as often measured by cortisol? Well, with prolonged stress, prolonged elevated cortisol levels, we see reduced volume in the prefrontal cortex and a reduction in synapses, or spaces where information transfer can happen between neurons, in the hippocampus. And these synapses would be important for the creation and consolidation of memories. Interestingly, most drugs that are classified as antidepressants can help to prevent this reduction of hippocampus activity and prefrontal cortex volume. In studying causal relationships, the diathesis stress model observes that you can have inherited vulnerabilities or predispositions towards certain problems or challenges, or what you inherit could be protections against certain problems, a kind of genetic or genetically bequeathed resilience. And that's your diathesis, your initial vulnerability. And this initial vulnerability or resilience is going to be what, in large part, determines the effect that any given stressor is going to have on you. So not everyone develops PTSD. Why? Well, some people have a diathesis or a vulnerability that others don't. So the same traumatic experience could have less traumatic effects on some people than it does on others. That's diathesis stress in a nutshell. But of course, even an apparently straightforward illustration of diathesis stress would tend to be complex. So this was a 2007 study in the American Journal of Psychiatry. They looked at the serotonin transporter genotypes of individuals who had all survived Hurricane Katrina. Now, some of these individuals developed PTSD and depression. Their question was whether those who had the short genotype would be more vulnerable to developing PTSD and depression. Is it the case that there were more individuals in the PTSD and depression bin with the short allele? Well, it actually turned out to depend. So we already have a diathesis. We have those with the short and those with the long allele. And we have the stress, which is Hurricane Katrina. But we need something a little bit more to explain what's in front of us. Because if people had adequate social support, then having the short allele did not increase their risk of having PTSD and depression. Social support protected those with and without the short allele diathesis. But if you had relatively low or no social support, then the short allele was associated with a more than 4 times increased risk of developing PTSD or depression. The short allele diathesis roughly doubled the risk of PTSD and depression compared to just having no social support. So the diathesis mattered, but its apparent risk was mitigated if the individual with the diathesis, with the short allele, had social support that they could draw from. So we have trauma and genes, or stress and diathesis. This model also includes the environment in which this trauma is occurring or being processed, and in which these genes are living. Finally, we had a question about genetics and schizophrenia, because you've likely seen an article like this from CNN, "Scientists confirm genetics of schizophrenia." Okay, so what does its genetic mean? What can this hedgehog tell us, and what can we as a fox realize is missing from this formulation? First, what is schizophrenia? A disorder of thought and emotion. You can either conceptualize it as losing contact with reality or having contact with non-reality. It's not common, but it is universal, so we would expect any population of humans that we come across, even if they've been isolated from the rest of humans for a long time, to have under, but around, 1% of their population experience this loss of contact with reality, or contact with some non-reality. Your hallmark symptoms include delusions, strong beliefs with no basis in reality. Usually they're impossible beliefs, but the DSM makes a distinction here, and it says that if the strongly held fixed belief is shared locally or culturally, then it's not to be considered a delusion. So if I think my neighbor is a witch, whether that is a delusion or not is determined by how many people around me would be welcoming of that type of idea. The other hallmark symptom would be hallucinations, hearing, smelling, feeling, or seeing something that is in fact not there. The most common form of hallucinations being speech hallucinations, hearing voices. Okay, but what does it take to get an actual diagnosis of schizophrenia? This is, after all, an abnormal psychology course. You must meet all of the letter criteria, and for criterion A, this includes 2 or more of the following, each present for a significant portion of time during a 1-month period, or less if successfully treated. At least one of these must be the first, the second, and the third of these numbered symptoms: delusions, hallucinations, disorganized speech, grossly disorganized or catatonic behavior, and negative symptoms. So if you have one of the first 3 and at least one other symptom, which could include another of the first 3, then you meet criterion A. You have to also meet the other lettered criteria. B is pretty much just adaptive failure. Are the symptoms from criterion A affecting your life negatively? Criterion C says you need to have been suffering for about 6 months or more. We have other diagnoses if the problem has not persisted for 6 months, but it's schizophrenia proper if it's been disturbing you for at least 6 months. Criterion D is just about alternate diagnoses, so if you've experienced depression at the same time as the psychosis of schizophrenia, then there are other diagnoses that may need to be ruled in. We also need to make sure your symptoms are not due to some substance that you might be exposed to or a medication. And F is just about ruling out other possibilities as well. So that's what it takes to get a diagnosis. Let's look at a case that's arguably typical. 7 months ago, Dave began to believe that his recently deceased friend may have faked his death using a twin. Now, opinions may differ on this, but I'm going to call this a prodromal mild delusion. In other words, it appears delusional, and I'm going to predict that the delusion is only going to grow and get worse. Note the color coding. I'm keeping track of the symptoms. Dave is gradually disengaged from social activities. This is what we call a negative symptom. What's symptomatic is what's not there. He's withdrawing from society, from his own previous normal, and he reports no motivation to see his friends. Another negative symptom, something that's not there, that should be or normally would be. He mumbles to himself, which in light of the other symptoms could be a prodromal indicator of disorganized speech, or something that might develop into disorganized speech. And he speaks in monotone. Note that Dave didn't used to speak in monotone. He's lost some of what we call his prosody, or prosody, which is just the way a voice tends to move up and down as it talks in order to keep you interested, to emphasize things, etc. Recently, Dave has begun claiming to see his deceased friend, quote-unquote disguised as other people, and despite showing some insight by acknowledging that others don't believe him, his belief has not changed. So if anybody was on the fence about delusion before, well, we can definitely check it off now. So conservatively, we have delusions and we have negative symptoms. In other words, we meet criterion A. We'll continue to get a case history, a social history, and it turns out that Dave has brought these symptoms into the world, i.e. his delusions have impacted the well-being of those around him and appear to be affecting his ability to function at work. We mentioned that this started 7 months ago. We rule out mood disorder, we rule out exogenous substances, and we rule out autism and communication disorders. And with that, we can diagnose Dave with schizophrenia. That's what it takes. Back to our question of where did Dave's problem, because it's only a disorder because it is creating a problem, where did this come from? Let's consult our biohedgehog, who is waving the headline from CNN in the air, saying, "I'll show you the evidence for why schizophrenia is all biological. Here it is. Look at the different risks that you have. Let's say your identical twin has schizophrenia. There's a 48% chance that you will have schizophrenia. Keep in mind that if you're just sampled randomly from the population, there'd be a less than 1% chance of you having schizophrenia. But if your identical twin has it, there's a 48% chance that you will have schizophrenia too. And this risk tends to go down with different degrees of genetic relation between you and the other person who has schizophrenia." Okay, thank you, Biohedgehog. Thank you for bringing visual aids. Psycho Hedgehog, what have you got? Well, it grows out of a personality trait, just like everything else. Those with schizotypal personality disorder are at increased risk for developing schizophrenia proper. And if Biohedgehog was right, and it's all genes, then why is it that most people diagnosed with schizophrenia have no family history of schizophrenia? That's a huge hole in the Biohedgehog's argument. Also, most people with the risk factors don't develop schizophrenia. Case in point, the Biohedgehog's own graph. If I have an identical twin with schizophrenia, I still only have a 48% chance of getting schizophrenia? It's clearly not just genes then, because I share 99.99 repeating percent of my genes with my identical twin. The only exceptions are replication errors. And if it's not just genetic, well then it's probably mostly an outgrowth of psychology and personality. Okay, but that might be a sort of false dichotomy, says our social hedgehog. Why not explain that which cannot be explained by genetics with the social sphere rather than the psychological sphere? My evidence, she says, that schizophrenia is all social includes that schizophrenia is more common in urban than rural environments. It's more common in minority ethnic communities, whatever the minority happens to be in the country that we're talking about. Schizophrenia is associated with birth complications, such as having periods where you couldn't breathe, and with things like stress, infection, malnutrition, and maternal diabetes. So at the very least, if we changed these aspects of society, stress, exposure to infection, umbilical cord strangulations, we would reduce schizophrenia rates. In fact, there's a Finnish study looking at children who had been adopted away from their mothers who had schizophrenia. They were interested in whether adopted away children whose mothers had schizophrenia were more likely to get schizophrenia themselves. And if the adopted family environment was quote-unquote non-disturbed, then there was in fact no additional risk for these adopted away children of schizophrenia. In other words, children who were adopted away from mothers with schizophrenia into stable family environments did not get schizophrenia at a rate higher than other children. Okay, social level hedgehog, you make a good case. Now I'm going to do my foxy thing and see how all these things combine. I'm going to keep in mind two things. First, all these things can be true at once. Perspectives, by virtue of being different, are not necessarily contradictory. So we're full circle. Hopefully that was somewhat enlightening. Stay tuned. We'll see you next lecture.